Okay, good morning. Nice to see you guys this morning, as always. We're going to have two lectures today. The first is on functional connectivity and what's called resting state fMRI. And the second lecture that I had planned for today was on DTI, diffusion tensor imaging. And as I was looking at it last night, I realized that maybe it was a little bit complicated. It's still even a little complicated for me. It involves a lot of linear algebra and eigenvalues, eigenvectors, and all kinds of concepts, diffusion concepts. So I thought maybe since tomorrow we only have 1 hour, that I would take a lot of the mathematics out of that. And I don't know if any of you have done tensor modeling or anything like that. So I figured I'd take that stuff out and we'll do a more conceptualized version of it. And hopefully we'll have time for a little bit tomorrow to talk about sleep. So today we're going to talk about this functional connectivity and resting state fMRI. And after that, we're going to talk about memory. And then, of course, we have the quiz or exam afterwards. So I will accompany you guys to there so we can see if anybody has any questions, and then we'll get started with that. You're welcome to use any of the lectures or notes. So this is why we're going to the computational modeling center? So you have access to all those things there. Okay. So functional connectivity, we talked about just briefly before. Functional connectivity refers to connections between different brain regions while a person is either resting or doing a cognitive task. So we talked a little bit before about how different areas of the brain work together to accomplish different functional or cognitive processes, including sensory information, reward processing, inhibition, emotional processing. And today we're going to look at this a little bit further and talk more about these connectivity issues. Now, functional localization refers to the approach that brain functions are specific to certain regions. And as we've seen, this is true to an extent, right? Different regions of the brain are responsible for different functional processes. However, when we talk about functional localization, we also need to consider what's called functional integration, which is the study of connected processes. So as I mentioned before, areas of the brain don't work independently of each other. So areas of the different brain talk to each other through these neural connections and integrate information and work together to make a complete process or do functionality in a complex way. So there's 3 types of connectivity I want to talk about today. The first is anatomical or structural connectivity. The second is functional connectivity, and this is functional connectivity we'll be talking about a little more extensively, and what's called effective connectivity. So anatomical connectivity refers to the process by which structures or functions are connected by the presence of axons. So these again are these white matter tracks, and we can assess this by examining connections that are already known to exist in the brain or by using diffusion tensor imaging methods. So like I mentioned, tomorrow we're going to talk briefly about diffusion tensor imaging, and diffusion tensor imaging look like the one here on the right. This is a structural imaging technique that employs magnetic resonance imaging and uses the fact that the diffusion of water takes place along the axonal tracks and in the main direction and doesn't really diffuse outside of the track. So we can take this property and use MRI to create these white matter tract images. And you can see here you get a very beautiful image which details the connections between regions of the brain. So that's diffusion tensor imaging and white matter tracting. and that's sometimes referred to as tractography. We also can look at connections that we know exist. For example, there is a known connection between Broca's area and Wernicke's area through this arcuate fasciculus. I'm not quite sure if I said the first word correctly, but it's a known group of neurons that connect these two areas. So there are anatomical connections in the brain that help with functional processing. Okay, the second thing we're going to talk about is functional connectivity. And we're going to talk about this even a little bit more today. And this refers to the fact that there's statistical dependencies between different regions of excuse me, of the brain that we can look at by doing a correlation analysis between different regions of the brain. And what we see here is when we do this, different regions act together or the same over time. So people have done a lot of work studying these types of functional connections and see that if we pick a region of the brain and do correlation analysis over time with different regions, we see that different parts of the brain that aren't connected anatomically are working together to accomplish a task. And like I mentioned, we're going to talk about these functional connections a little bit more. So these are typically done by, again, picking a seed region, what's called a seed region in the brain, and looking at time correlations between this region and other regions in the brain. And again, we can see that they're working together. So they can either be working together concordantly, so as one is going up and down, the other is going up and down in time. So these are regions where we're seeing activity in the brain, and the two regions are acting similarly together. And they can also be working with a negative correlation. So one region of the brain is going up and down while the other is going down and up, which is shown here in this plot to the bottom right. So you can see the blue line is going up and down while the orange part is going down and up at the same time. So we say this is a negative correlation between the two brain regions, but we can see that they're still acting together in a time-locked type of manner. Effective connectivity is causal or directed influences from one region or group of neurons to another. So what this means is that if one region of the brain activates, it has an effect or a direct influence on a group of neurons in another region of the brain. And we see this sometimes in the frontal lobe and its interactions with other parts of the brain. So the frontal lobe sometimes will have a process that can come back and inhibit in a part of the more primitive center of the brain. So we call this effective connectivity. And this depends on the functional interactions between, again, one region of the brain and the other. Okay, resting state functional MRI is a little bit different than the functional MRI that we've been talking about, because this is functional MRI that's collected while an individual is not performing an explicit cognitive task. So this is sort of background activity in the brain, and this is a fairly new method that people have been focusing on more, because when we initially started doing fMRI, we knew that to see a difference in blood flow in regions of the brain for a certain task, we need to alternate between two states, right? If you're going to see a difference in something, you need two states to compute that difference. So resting state fMRI wasn't really a thing when it first came around. But then as people collected more and more fMRI data, they started to notice this background activity that we're going to talk about a little bit more that reminds me a little bit of the alpha activity of EEG. So do you remember the alpha waves of EEG are present when somebody is resting with their eyes closed? And what people were seeing in the fMRI data was something similar. There was some kind of background activity that people noticed. So the brain regions that are temporarily correlated during this resting state are called the default mode network of the brain. And again, we're going to talk about this a little more. So let's look at an example of a task-evoked fMRI paradigm. By task-evoked, we mean that there's two different states, and somebody is evoking a change in the blood flow in their brain due to these different states. So we have a person alternating between eyes open and eyes closed. So the bold changes from eyes open to eyes closed are apparent. You can see here on the bottom left plot when the regions in the occipital lobe are exposed to the open eyes, they go up, and when they're exposed to the closed eyes, they go down. So this bold activity is oscillating up and down with the task. But what they also noticed was that if they took this oscillation out, we still had this background noise activity. So you can see the higher frequency waves that are occurring up and down as the person goes back and forth through this task. So they asked the question, what actually is this activity? What is this thing that we have considered to this point to be noise? And what they thought they were seeing was some kind of a spontaneous bold activity. So an up and down oscillation of the signal in the absence of a cognitive task. So again, the brain is always active, even in the absence of explicit input or output. So this the task-related changes in neuronal metabolism are only accountable for about 5% of the brain's total energy consumption. So what is the brain doing what is it using its energy towards if it's only using a small amount to do some kind of cognitive task? So another way to phrase this question is, what is this other oscillations, or what is this signal that we're considering to be as noise in this functional activity that we're measuring by using the bold signal? Okay, so what they attributed this, again, this was called the spontaneous bold activity. It occurs when if somebody is doing a task and while somebody is at rest. So this is intrinsic brain activity. And this is sort of intrinsic, meaning that it's going on in the background. And resting state networks are, again, correlations between this spontaneous bold activity when we look at different regions of the brain. So they noticed that different regions of the brain were exhibiting this spontaneous activity at the same frequency or in connection with each other. So as people looked a little bit more into this, and again, you can pick a seed region of the brain and take this up and down oscillation, God bless you, and you can correlate all the other regions of the brain to it. So you were looking at dependencies that are sort of locked together during time. They're moving up and down together over time. And what they found were these different resting state networks. Okay, so this was taken from a paper that discussed MDD, which is major depressive disorder, and what I want to focus here on more is these networks that they're talking about. So the first one on the top row here is called the default mode network. So it involves the regions that you see here highlighted in red. And this default mode network is thought to be this network that takes place in the background. And this is particularly going on while a person is not doing an explicit task. So we're going to look at this a little bit more detail as we go on through this talk. They also saw this what's called the central executive network, and this network they saw was going on in the background while a person was thinking or doing a task. So while a person is doing a task, you can look at the background up and down noise and do a correlation and analysis across time. And they were seeing that these blue networks were lighting up. And the final one that we're going to talk about in a little more detail is what's called the Salience Network. And the Salience Network, they noticed, was sort of the network that keeps us attentive to what's going on in our surroundings in the background while we're doing other things. So we're going to talk about each of these a little bit more. The default mode network is thought to be activated or oscillating in the background when a person is not involved in any sort of cognitive task. So this is active when a person is awake and not focused on the outside world. So they're doing something such as daydreaming or mind wandering or thinking things to themselves. So there were some different things that people could do that they noticed this default node network was active. And so we're going to talk about each of these things. The first was that they noticed that this default mode network was active when somebody was thinking about themselves. So they were pondering maybe on autobiographical information, recalling memories or events that have happened to you in the past. They were self-referencing, so they were thinking about traits or descriptions about themselves. They were emotionally regulating, so they're reflecting on their own emotional state. So we all have these moments when we're sitting quietly and pondering about ourselves and thinking whatever we think about ourselves. And while people were doing this, they noticed that this is one time when this default mode network was active. They also noticed that this default mode network was active while people were thinking about others. So theory of the mind, thinking about the thoughts of others and what they might or what they might not know. So if I'm looking at you and thinking, I wonder if they're absorbing this information or if I'm presenting it in a way where it's too difficult for everybody or not, and I'm sitting quietly thinking this, my default mode network may be active. They also noticed it was active while people were thinking about the emotions of others. So trying to understand the emotions of other people and empathizing with their feelings. It was activated during moral reasoning, or so when somebody was determining just and unjust results of an action, and when people were doing social evaluations. So while they were having a good or bad attitude and judgments about social concepts in social categories. So this would be reflecting on important social characteristics and statuses of a certain group. So again, this network is active while you're thinking quietly about yourself, thinking about others, and also while you're thinking about memories and thinking about possibilities of the future. So this network could be activated if you're thinking about the past or recalling events that happened to you in the past, imagining the future, so thinking about things that might happen in the future, about maybe plans that you have or dreams that you have for yourself or others. In episodic memory, so when you're thinking about memories related to specific events in time, or story comprehension, understanding and remembering a narrative. So there's a lot of times when this default mode network is active in the brain, and it's usually while somebody is doing some quiet reflections. Okay, so I showed you in that figure of these brain networks a second network called the central executive network. And this is the brain network responsible for high-level cognitive functions, notably the control of attention and working memory. So this part of this network of the brain seems to activate while people are doing active tasks. So this is the noise background that they saw while they were having people do cognitive tasks, and the person had to pay attention and process this information. So not only were they activating these task areas, but if you looked at those background oscillations and ran a correlation to see which voxels in the head had this background oscillation while a person was actively doing a task, that's when they noticed the central executive network was turning on. So activity in the central executive network correlates with performance on executive control tasks. So when you're trying to focus, when you're trying to think, your frontal lobe is sort of doing this executive functioning of keeping you attentive, having your working memory employed if you need it for certain things, and activating in concordance with the regions that were responsible for the cognitive activity. So another interesting thing about this network is that the strength of the within network connectivity in this central executive network is also been found to be associated with higher IQ in children, adolescents, and adults. So people who tended to be smarter or have a higher IQ had more of this background activity going on while they were doing cognitive tasks. And they also noticed that it was anti-correlated, so this is a negative correlation, with activity in the default mode network in healthy adults, and it may even inhibit the default mode network under certain circumstances. So you have this default mode network going on when you're relaxed, then someone asks you to do a problem or do a memory task. This default mode network turns off a little bit and the central executive network goes on. So again, when one network is becoming active, the other is becoming more inactive. The third network that I showed you on the plot was what's called the Salience Network. So this is a network that attends to importance of matters. So we're always getting this sensory information from all of our senses, and it's constantly going on. And fortunately, our brain is able to filter out what is important and what is not. So this is part of the Salience Network, the ability to select things out of the environment that are salient or important to us in some way. So they're notable or they stand out, they're meaningful in some way, or they're behaviorally relevant to what's going on for us personally. So this is thought to also regulate changes in other networks, right? Because if you need to instantly pay attention to something, then you say we heard a loud noise or some kind of explosion, we would all come out of our default mode network. We would stop thinking about whatever is going on in our own brains and all immediately pay attention to what was going on. And we may be doing math problems or something and have this cognitive network going on. Again, if there was an explosion or something that got our attention, that would also turn off, and we would know due to the Salience Network that we need to immediately pay attention to the important things going on in our environment. So this is thought to mediate, again, a response to important internal or external signals. So internally, meaning, say you got a really bad stomach ache all of a sudden, this would also get our attention and maybe pull us out of some kind of deep thoughts about ourselves or our intense math problems that we were working on. Okay, so these resting state fMRIs are collected by having a person lie still and not doing anything at all. So we're trying to measure this time course, again, of the spontaneous bold response that's going on while a person is not doing anything. And as you can imagine, this spontaneous activity is less susceptible to task-related confounds. So what this means is that, as I mentioned before, we might have somebody who's moving a little bit to a task or having other influences that are happening in their brain that are related to the task but not due to the task itself. So these are confounds in the brain that are going on throughout the experiment. But when you just have somebody resting quietly, you don't have as many of these problems going on. So in our regular fMRI experiments, again, we had two different states that we alternated, and we looked at differences in the states. So again, how are we going to measure which regions are active while this spontaneous bold is going on? And as I mentioned before, we do this with a seed voxel, and we do this by taking correlations with this seed voxel in different voxels in the brain over time. So here we go as an example. A shows this resting state fMRI that we might be doing. So we're not doing anything over time. We're just sitting there quietly and letting these scans take place. You could also get a resting state out of a task that might involve the finger tapping versus rest. So the bottom line here is that we're getting a seed voxel during rest, whether it be during the whole time of rest or we want to pull the rest out of a task that we have alternated with. Then we take this seed voxel and we run correlations with it through all the other voxels in the brain. So again, we're looking at how locked in time two parts of the brain are oscillating with each other or oscillating against each other. So they have to be either positively or negatively correlated. And again, correlated means that they're working together over time. And then we can see which regions are working together by taking the regions that are correlated, highly correlated, and we can get this functional connectivity map. So this is how people have determined how these networks, where they are, and then make proposals about how they are working together, depending on what the person was doing while they were in these oscillating states. Is that making sense to everybody? Oops. Okay, so again, pick a seed region, correlate the time series of this seed with the time series of all the other voxels, and we can see where in the brain these are time locked with each other or working together. So I gave you the three major networks, but people also are finding more and more functional networks all the time. So if you look at each of these, the black dot is where a seed region was taken, and more than 1,000 subjects were used to compute correlations between these seed regions over time, and they found these other sort of networks. So again, the executive control network can be shown here on the left, and this is active while a person is doing a task. This is a background activity active while a person's doing a task. The default mode network is down here on the bottom left, and we talked a bit about that. The Salience Network here is directly on the bottom, and they also found these visual networks, sensory motor networks, and dorsal attention networks. And they put these all together, and you can see that this covers a lot of the brain if you account for networks that are going on in all the different areas. So I think this is just really exciting and interesting research, and it's one of the steps that's used to try to find out what the brain is doing in the background, what it's using all this energy for if it's not explicitly doing a task. So you hear people say that we really only use about 10% of our brain, and I'm not sure if that statement is true. I think our brain is doing a lot of things that we really don't understand fully while we're engaged in whatever activities that we're doing at the time. So we always have these background processes that are active and keeping us safe and attentive in our environments. So we can examine differences in connectivity in patient populations with particularly clinical problems and see how maybe a different or a clinical problem would affect these networks. So one thing that they've looked at is ADHD, which is this attention deficit hyperactivity disorder. And they found that kids with this disorder typically have reduced connectivity within the default mode network, and reduced anti-correlations with the default mode network, and increased connectivity in the Salience Network. The Salience Network, again, is something that's just grabbing our attention and making us focus on other things. So this would make sense that somebody with attention deficit hyperactivity disorder may be overstimulated by salience or things that are going on in the background, and they may have less attentiveness to things going on internally because they're overly stimulated by things that are going on around them. They've also looked at healthy aging populations and saw that there's decreased correlations within the default mode network as you age. And they also looked at Alzheimer's patients, and they saw that there are decreased correlations within the default mode network, including the two hippocampi in the brain, the left and right, decreased anti-correlations with the default mode network, and reduced local connectivity as reflected in clustering coefficients. So one thing about Alzheimer's disease that is interesting is that when I worked in this area, it was thought that the parts of the brain that start to sort of decay are parts of the brain that are directly associated with this default mode network. So people have looked at this default mode network more in Alzheimer's disease, and specifically in inherited Alzheimer's disease, which is sometimes an early onset Alzheimer's disease. This is a form of Alzheimer's disease that develops before the age of 65. And when I worked at the PET Center at the University of Pittsburgh, we saw people coming in with early onset Alzheimer's disease as young as in their late 40s, which is really just really striking and a very sad situation, especially for family members and for the person themselves. So this is thought, this genetic Alzheimer's disease is thought to be caused by these different genes that you can see here in the middle. I'm not really a genetics person, but I know that if you have certain risks for things, sometimes they can measure it in your genes. But what they saw was adults with this dominant Alzheimer's disease mutated gene showed decrease in connectivity throughout the default mode network relative to non-mutation carriers, so relative to people who didn't have this mutation. So, and they looked at this using functional magnetic resonance imaging in these resting state networks. So it's been proposed that one of the problems with Alzheimer's disease, or one of the symptoms, is that there's a problem in this default mode network of the brain in Alzheimer's disease. Okay, so in summary, functional connectivity of fMRI can be used to show regions that work together in the brain. These regions may not necessarily be connected anatomically, but we're seeing that their spontaneous activity that we can measure is locked together in correlations over time. So they found that several connectivity networks exist, and more are being found all the time. Currently, this default mode network is the most popular one. It was really the first one to be found, and is one that people have focused a lot of attention on. And connectivity analysis can be used to examine neurological disorders in the hopes of understanding what the underlying mechanisms are of the disease. Again, like in the case of Alzheimer's disease. So that's all I have on this functional connectivity talk. I know we have the exam today, so maybe we'll take a little break early, and then we'll come back and we'll talk about memory, and then we'll head over to the testing center. Okay? Does anybody have any questions? Okay, everybody ready? We're going to get started talking about learning and memory. As I mentioned earlier in the class, I was initially going to talk about diffusion tensor imaging, And as I was going through my lecture last night, I realized how complicated it was. It uses a lot of linear algebra, um, tensor analysis, and it was a long lecture, very computationally intensive. And so, because we're running so short on time, I thought I would do the learning and memory lecture today and go back to that diffusion tensor imaging lecture, take out some of that complex mathematics because at times it's hard for even for me to understand. So, and we'll just talk briefly about the method and what it is and what it shows instead. And hopefully tomorrow we'll be able to talk a little bit about sleep. So we have a 1 hour lecture tomorrow, so I'm going to try to just do a short part of each of those tomorrow. And then we'll wrap up. Unfortunately, bless you. Unfortunately, these past 2 weeks have gone so quickly, and it'll be sad to leave all of you. I'm gonna give you my email address and contact information in the U.S., and I hope you will all keep in touch with me. And if you ever want to come to the U.S. and see the labs about brains in our MRI center, I would be very, very happy to host you when you came over. So I hope you'll all keep in touch. Anyways, aside from that, we're going to talk a little bit about learning and memory. This isn't an area where I research a lot, so but it is that one that really fascinates me. So we're going to talk a little bit about what memory and learning is, the types of memories, how memories are formed, and where in the brain these memories are located or where they're formed. And this is more of what parts of the brain are used in these processes, not necessarily where they're stored, because this is still a mystery in neuroscience research. So how a memory can be maintained and improved. And I know we've talked a lot about Alzheimer's disease. We're going to touch on this again because when you talk about memory, that's probably one of the biggest memory disorders that affects our society today. So what is memory? Well, memory is our ability to encode, store, retain, and subsequently recall information and past experiences. So we're exposed to something happens to us, somehow we store this in our brain, and we can remember this, and we can pull this information out and use it to affect or influence our current behavior. So it's thought to be in general terms as the use of past experience to affect or influence our current behavior. So if you have a bad memory of something, maybe you won't try to encounter that situation again. On the other hand, if something made you very happy and made your reward system feel very excited and activated, then you may want to try to do that experience again. So what's the difference between memory and learning? While memory is related to but distinct from learning, learning is a process by which we acquire knowledge of the world and modify our subsequent behavior based on this knowledge. And during learning, it's thought that neurons that fire together or that neurons that work together produce a particular experience that are altered so they have a tendency to fire together again. So to put this in other words, if there are neurons that fire together due to a process that you're learning, you'll strengthen this connection between these neurons as you learn, and then this information is thought to be reinforced by that stronger development of these networks in the brain. So we're going to talk a bit about types of memory, and we're going to talk about long-term versus short-term, or short-term is also called working memory, versus sensory memory, which we're going to talk about a little bit. Declarative versus non-declarative, episodic versus factual. We also have things such as emotional memories and fear memories. And we saw this a little bit when we looked at that video on trauma about how memories can work or not work during fearful or traumatic events. Okay, short-term versus long-term memory versus the sensory register. So we're constantly registering sensory information as it's coming in from all around us, but this information is quickly lost. We don't hold on to these memories or all the sensations going on around us. If our brain tried to keep these all the time, I think we would be pretty overwhelmed with sensory information. So things that aren't important to us or don't get our attention are quickly lost. And then we come to this, things that do get our attention can be held in a short-term storage. The short-term memory can hold about 7 items for about 20 to 30 seconds. So if I asked you to memorize a series of numbers, you may be able to hold on to this information for about 20 to 30 seconds. And if you don't continue to rehearse these numbers again and again in your head or devise some kind of tactic to hold on to this information, it'll be lost. On the other hand, if you are something that you go over and over again in your head, maybe somebody's phone number that is very special to you or something, you can develop this into a long-term memory. And there are different ways to do this. So one example might be you dial this number 2 times a day, every day. After a while, this is going to encode into your long-term storage. So again, short-term memory information can be gradually transferred to long-term memory if it's something that is important or relevant to us. Okay, again, short-term memory corresponds approximately to the initial 200 to 500 milliseconds after an item is perceived. Short-term memory allows recall for a period of several seconds to a minute without rehearsal. So if I told you to memorize 15239, you may be able to recall this to me in a few seconds, but if you don't keep this or practice it or try to remember it purposely in your head, and I just say, hey, what were those numbers, you may not be able to recall those because your short-term memory has a limited capacity, can only store 4 to 5 items, and if it's not rehearsed, it's information that's quickly gone. There are different ways you can try to increase your short-term memory, and for example, a process called chunking. In recalling a 10-digit telephone number, a person could chunk the digits into a group of 3. And in the US, we have area code, which is 3 numbers. Then the first part is 3 numbers, and the last part is 4 numbers. So you could try to remember these as chunks of information, or if it's something easy to remember, like 1, 2, 3, 4, 5, 6, 7, 8, 9, that's an easy way to recall. So there are different strategies and different ways that we use short-term memory, and sometimes we do this without even thinking about it, employing these strategies to help us remember things. Short-term memory is thought to take place and to be held in the prefrontal lobe of the cortex. So you can see there, again, the frontal lobe is the blue part, and at the very front of the frontal lobe is this prefrontal cortex, which again is thought to be responsible for executive function, higher order processing, and also for short-term memory. Long-term memory is something that can store much larger quantities of information for a potentially unlimited duration of time. And this can span to sometimes a whole lifetime. Its capacity is immeasurable. So we don't really know if there is a limit to long-term memory. We don't think so, but I don't know how we could find this out. Just as far as we know, there's no limit, and there's, as I mentioned, you can store information for a whole lifespan. I'm sure all of us can recall memories that we had as a child that were good or maybe even bad, but left a mark on us and is ingrained in our memory or long-term memory. So these memories are maintained by more stable and permanent changes in neural connections that are widely spread throughout the brain. So we don't know a whole lot about how long-term memory is encoded, but we do know that the hippocampus is essential for learning new information and in the consolidation of information from short-term to long-term memory. Although it's thought that the hippocampus itself does not seem to be a storage center for memory information. So the hippocampus may be involved in changing neural connections for a period of 3 months or more after the initial learning took place. So you may be, what this means basically is that it may take a period of even 3 months to really ingrain something into the long-term memory system of the brain. There are several types of long-term memories. We're going to talk in detail about each of these a little bit. And each of these long-term memories may use multiple brain systems for its processing. Okay, so the first type is a cognitive or declarative memory. These are the two basic categories. The other is non-declarative memory. So declarative memory is explicit facts and events. So this is information that is stored and retrieved, memories that can be consciously recalled. For example, history lesson, facts, dates. So if you're memorizing information for an exam, you may be using this declarative memory. Well, hopefully, if you have to memorize a list of facts, then you have them in your long-term memory. When you see them on your exam, you say, I remember 1492, Columbus sailed the ocean blue, or something like that. So that's a U.S. history thing there. And so I think you all get the point. It's just facts that you remember and have to recall or can consciously recall by thinking about them. Non-declarative memory, on the other hand, are implicit skills, habits, emotional, skeletal memories. And these do not require conscious thought to recall. So a procedural memory, for example, driving a car or tying your shoe. So the first couple times you do this, you may have to think about it, but after a while, you just get the skills to do this over and over again effectively without having to sit and think about it or recall it. Okay, so this is a classification bar of these memory systems, or a diagram. So again, we have the short-term memory, or it's also called the working memory, and we have the long-term memory. Again, long-term memory can be divided into declarative or explicit, or non-declarative or implicit. And what underneath each of these types of memories is a little schematic about what part of the brain is thought to be employed during these memories. So episodic memories and declarative would again include personal episodes in time or space, things that happen to us, recalling of memories, or semantic memories. So this would be facts, meanings, concepts, and knowledge about the external world. And these types of memories are thought to be, again, related to the hippocampus and the medial temporal lobe and the neocortex. The neocortex, again, being the cerebrum, or that new part of the brain that we use, new compared to evolution throughout history, compared to different types of animals. Semantic memories are thought to be in the lateral and anterior temporal cortex, and also in the prefrontal cortex. So these non-declarative memories, again, there's some different examples here than what we just talked about. Procedural memories, which would be skills and habits. Again, driving a car, tying your shoe, things that you learn to do through practice and procedure and come easily to you with practice and learning. There's priming and perceptual learning. There's the simple classical conditioning, and there's also non-associative learning. So we're going to look a little bit about what each of these are. Okay, this is a type of memory called priming. Priming is when an implicit memory effect where exposure to a stimulus is the response to a later stimulus. So for example, here we read the words night, moon, and sleep. And then we come to the bottom here, and what is the following word? What would you say that word is? Dark. Very good. Because we are influenced by the words that we just read, night, moon, and sleep. So what if those words instead were mathematics, geek, and extremely smart? We might say that word is dork instead of dark, right? So this is an example of priming, how something that we see makes an influence on something that comes next without even us knowing about it. So it just happens instinctually. Another type of memory that's a non-declarative memory is called perceptual learning. Perceptual learning is a process by which the ability of sensory systems to respond to a stimulus is improved through experience. So an example of this would be a radiologist who looks at pictures of brains every day. And they might be able to look at a picture of a brain and see a small tumor in the brain very quickly compared to somebody who doesn't look at these pictures every day. So this is perceptual learning, or being able to pick out more detail in a stimulus because we've been exposed to it over and over again. So another example I saw about this was maybe a dog show judge. So in the US, I don't know if they have them here, but people have their dogs. They brush them and fluff them all up and put bows in their hairs. They march them in front of judges, and the judges pick out which is the best dog, and they win the prize. So we look at the dogs and we say, those dogs all look the same. How do they know which dog is the best? Well, because they've looked at so many, and their sensory information on this has increased over time, so they're able to tell more distinctions in something that we wouldn't see distinctions in. Do you guys have dog shows here in India? You do? So yeah, it's interesting. It's kind of cute to watch on TV sometimes. So another type of memory is classical conditioning. This is a non-declarative memory again, something that we don't purposely store and then recall again. And this is the classical example of Pavlov's dog. I know we talked about this in an earlier class, and I couldn't remember the exact specific details of it, but here it is, okay? So before conditioning, Pavlov would bring his food to his dog, and his dog would salivate. The food was the unconditioned stimulus, and the salivation was his unconditioned response. So he also took a bell and he rang it. This was his neutral stimulus, and his dog didn't have any salivation, okay? But then he did this conditioning with his dog, classical conditioning. Every time he presented his dog with the food, he rang the bell. So the dog learned to associate that bell with his food, so that when he rang the bell, even without the food, the dog would start to salivate. So this is an example of how taking a conditioned stimulus, that being the bell, and pairing it with an unconditioned response, or the salivation, so that when you have this conditioned stimulus in the absence of your unconditioned stimulus, the food, you get this conditioned response now. So classical conditioning is sometimes takes place in fear learning, when you're exposed to something repeatedly that caused you trauma, and you have this trauma event again in the absence of the danger presence, but just something that reminded you of that experience causes this fear response in you without the actual danger of it being there. And this is something that's thought to happen in PTSD, post-traumatic stress disorder. Okay, another type of memory is this non-associative learning. Non-associative learning refers to a relatively permanent change in the strength of response to a single stimulus due to repeated exposure to that stimulus. So this is also called habituation or desensitization. So say that you were exposed to a loud noise over and over again, and this noise initially startled you very much and you had a big jump to this noise. Well, if you heard it over and over again, you may not jump to the noise anymore. So one example I can think of this too is in driving here in Pune. So everybody's beeping the horns, and when we first got here, we were like, oh my goodness, people are beeping everywhere. The US people don't beep quite as much. So if somebody beeps at you, you immediately say, what did I do wrong? Or you think there's a problem. But after you're exposed to people beeping all the time, you sort of become desensitized to it. So these events don't startle you or cause the same reaction in you anymore. Okay, we're going to watch this video of the case of HM. So HM was a unique subject, and they used his initials for a very long time due to privacy purposes. But HM was a guy who had basically a type of amnesia where he could not form new memories. So he had a surgery when he was in his 20s that removed his hippocampus because he had seizures, and this was a more common thing to do back in the first half of the 20th century, and he could no longer form new memories. So this is just a little video about him and the things that they learned. He was a very classic case of studying about memory for psychologists and psychiatrists in the US.
HM was one of the most fascinating patients in psychological history. He suffered from anterograde amnesia after having surgery in 1953. His name was Henry Mollison. He was born on the 26th of February 1926 and he died on the 2nd of December 2008. Here's a brain scan of a normal brain on the left and HM's brain on the right. You'll notice the hippocampus has been removed. This was to deal with his intractable epilepsy. It was age 27 on August 25th, 1953, when he underwent the surgery. This involved removing the hippocampus, and most of the amygdaloid complex and the entorhinal complex was also removed, and some other areas were damaged, but this is not known if the surgery itself did that or the brain later changed, because some of this was from the 2009 study of his brain. HM had trouble forming new explicit memories. These are memories which you can consciously bring to the surface. So for example, I know that France is north of Spain. HM would be able to tell me that after I told him that, but he would not be able to remember it long term unless he already knew it prior to the accident. That's working memory. So if I was to tell him 5 digits, 7, 9, 6, 3, 4, he would be able to recall those digits to me. And his procedural memory was also intact. This is where he could learn new motor skills, but he would not be able to remember learning the new motor skills. So for example, I'll give one actually at the end. His IQ was in the normal range. He couldn't remember anyone he met after the surgery, even people he saw every day for 20 years. He'd always meet them as if it was the first time he'd ever met that person, even if that person already knew them. The mirror trace task. This is the example of the procedural memory. After many trials with the mirror trace task, which is this, he got better, but he couldn't remember previous trials. This suggested both a difference between types of memory and different areas of the brain that are responsible for those types. HM was shown a picture of himself in the mirror. This is when he was an old man. He then became upset because he didn't realise he was an old man. He had the surgery when he was 27 years old. When he was an old man looking at himself in the mirror, he hadn't realised that he was an old man. He still thought he was 27 because he can't form new memories. Interestingly, he actually forgot that he was an old man 15 minutes later.
So the mirror trace task that they mentioned in there is a task where you have to trace a star by looking in the mirror. And the difficulty with this task is in the mirror, when you put your hand in one direction, you actually see it go the other way. So you have to learn this task by doing it over and over again to get better at it. In the case of HM, he got better at this as he did it daily, but he had no memory of ever doing this task before. So that was very interesting. So if you could remember what they said, he could do his short-term memory, his procedural memory, and some of his memory tactics were in place, but his explicit memory and the ability to remember people was highly compromised. So he could meet the same people every day for 20 years and never even realize that he met them. So it's really hard for us to imagine, I know. So it was a very interesting case. So if you're interested in this more, there are a lot of videos online about him, including one from the son of the surgeon who did his initial surgery, taking out his hippocampus. So how do we form memories? So how do we create long-term memories, and where do we store them? Well, this is a good question that people are researching all the time. And one of the primary functions of sleep is thought to be improving consolidation of information. Several studies have demonstrated that memory depends on getting sufficient sleep between training and testing. So this is important to remember if you're studying for a big exam. If you study and don't get a lot of sleep, you may not have these memories locked well into place. So the best practice would be to study a little bit every day, and as you sleep, hopefully you're locking these memories in to storage. And they've seen cases of this with, for example, people who have experienced traumas and then don't sleep. They seem to do better than people who have experienced trauma and then sleep for a while, because again, the sleep is thought to be what's locking into place these memories. So data obtained from neuroimaging studies have shown that activation patterns in the sleeping brain mirror those recorded during the learning of tasks during the day, suggesting that these new memories are solidified through rehearsal in the brain. This is particularly when you're in rapid eye movement or REM sleep. It's thought that you're rehearsing these in your brain and helping them to go into long-term memory. So one mechanism that is thought to be involved with learning and memory is synaptic plasticity. So this is the ability of neurons to change their synaptic connections over time. And if you remember, the synapse was that junction between two neurons. So this is the process between which one neuron sends information to the next is also called neurotransmission. So these connections can strengthen or weaken over time depending on learning processes or repeated exposure to something. So connections are changed by either increasing or decreasing the quantity of neurotransmitter in the synapse. The adaptation of neurons due to learning and memory is called Hebbian Theory, and this was proposed by Donald Hebb in 1949. So this was pretty early on that he proposed that neurons themselves can adapt, and these synaptic connections can change over time in response to learning or adaptation to something. So there's two mechanisms that are, one is termed long-term potentiation and one is termed long-term depression. If you look at these on the web, you will see that these can be pretty complicated processes, but the basics of it is that forms of synaptic plasticity are these long-term potentiation and depression. So these are forms of synaptic plasticity. These, again, are changes in the brain that support learning and memory. So long-term potentiation, or LTP, is a persistent strengthening of synapses based on recent patterns of activity. So again, as you continue to use or repeated exposure to something, you're strengthening patterns of synapses based on what is working in the brain in response to this task. Long-term depression, or LTP, on the other hand, is an activity-dependent reduction in the efficacy of neurosynapses, lasting hours or longer following a long pattern stimulus. So this is the loss of the, or sort of the decrease in the strength of a synapse over time. So these two mechanisms are in place to help us learn or get rid of things that are not relevant to us. So again, where is memory formed? So or where is memory? So we saw before a little briefly about where they thought each type of memory process took place. But one thing we know is that the hippocampus is very important in the formation of memories, and particularly in the formation of declarative memories, or those memories that we store and recall upon demand or need. So memories are not stored in the hippocampus. The memories are thought to be distributed throughout the brain, but this hippocampus is a major player in consolidating and helping us to store these memories. So again, the type of memory that it is or the location of where it is dependent on the type of memory that it is. And here's thought to be, again, where some of the different types of memory locations take place. So the procedural memory is thought to take place in the striatum and putamen deep inside the brain, and there's tracts of neurons that are thought to be associated with dopamine. Emotional memories could take place in the hippocampus, and these, again, we remember that the amygdala, I'm sorry, I said hippocampus, but I meant amygdala. So we remember that the amygdala is involved in emotional processing, and it's thought that this is also where the fear learning or fear memories are thought to be associated with. So many regions of the cortex are responsible for short and long-term semantic and episodic memory, and it's thought that the cerebellum is involved with procedural memory, or those memories that we learn from repetitive doing over and over again. The medial temporal lobe, again in the hippocampus, are important for long-term semantic and episodic memory. Okay, so you might ask, how can I improve my memory? So I'm going to give 5 simple techniques that I found that can help in strengthening our memory or improving our memory. So one of these techniques is called chunking. So chunking is the organization of material as meaningful, so that you can think out relationships among each group. This is not only improves learnability, but retention, and helps aid in faster and effortless recall. So limit the chunks to 5 to 9 items, and this is, again, this partly due to the short-term memory capacity. And an example of this might be, okay, you have the following list when you go to the grocery store or out to the store. You need to get apples, cucumbers, paper, ink, cabbage, banana, grapes, beans, a stapler, and an orange. Okay, so if you don't have a piece of paper and you want to try to remember this list, you could rearrange things into, so you might start with the fruits that you need: apple, banana, grapes, and orange. And remember that you need 4 fruits. Okay, then you could go on and remember you need 3 vegetables. You need cucumber, cabbage, and beans. And then you need 3 items of stationery or paper related items, being your paper, your ink, and your stapler. So instead of trying to remember each item in this list as its own sort of entity in itself, you can chunk this information together and remember I need 4 fruits, 3 vegetables, and 3 stationary items. And then this will help you store that better and recall it then when you get out to the store and start shopping for what you need. Okay, rhyming is another good way to remember. And it's one of the most popular and oldest methods in memorization. So this technique makes use of the fact that we have a natural tendency to remember rhymes and rhythms. And here's an example of what we might say. 30 days hath September, April, June, and November. Once short February's done, all the rest have 31. So this might be a rhyming technique that we can employ to remember which days of the month have 30 days versus 31. And of course, February is its own unique short month. Another thing of this is with songs. So sometimes we'll remember song lyrics because they have a tune along with them, sometimes a rhyme and sometimes a rhythm. And I remember when we were in our biology class, there was a professor who wrote a song about the Krebs cycle, and he would sing it and play it on his guitar so that people could learn it and remember all the steps in the glycolysis or the Krebs cycle. So that was interesting. So if you ever have to remember something that you don't have a piece of paper, make up a little tune about it, and then you can maybe try to remember it that way. Okay, another form of memory that you improving a technique that you can use to help improve memory is mediation or bridging. So in this method, a bridge is built in between items given to be memorized. So this technique is best suited for learning material involving word pairs or material that can be reduced to word pairs. So one example of this is learning the capital of Poland. So the capital of Poland is Warsaw. So in World War II, the beginning of this started with Germany's attack on Poland. Thus, it could be arranged as Poland saw war first. And then you could remember Warsaw based on this little phrase that you came up with. So this is, again, bridging or developing a mediation between two events that were related to help you pair these up. Okay, this is a great way to remember things, I think, is a bedtime recital. So in this technique, you do your recital or rote learning just before going to bed. Then your mind is in the process of sleeping. It could arrange the information as a systematic and effective way of remembering it. So this again has to do with memory consolidation as you're sleeping. So psychologists have found that if you sleep after thinking about your problems, there's a better chance that you arrive at a solution the next day. So a friend of mine always said, don't worry at night. If at night your problems seem big and looming, get a good night's sleep and think about them the next day. So this is also maybe true with memory. So steps for a memory improvement or be in a relaxed mood. Write down the things you're supposed to remember on a piece of paper and read it aloud, if possible, once or twice. Recite it 2 or 3 times. Then go to sleep without worrying or thinking about it instead of sitting up, maybe stressing about a test all night. And maybe you'll recall it or find it easier to recall when you're not focused on it. So sometimes this happens too when you're trying to remember somebody's name or trying to remember something and you think it's on the tip of my tongue, I just can't bring it to my forefront, and you think, okay, I'm just not going to think about it for a while, then it'll pop right into your head at one point when you're doing something completely different, right? So this is because you're not putting stress on trying to remember, you're feeling more relaxed. So I think this bedtime recital too, as I went through school and got into more complicated subjects and had to study, I found that studying maybe even for 20 minutes a night before going to bed on the same topic was a much more effective technique then waiting till the day before and cramming all the information, getting very little sleep, and then trying to recall it the next day. It took me a couple of years, though, to to work out that that's a lot better technique to use. Okay, trying by not trying. Okay, so this is actually what I just described. Sorry. All of us apply this method knowingly or unknowingly. Sometimes when you try to recall, you may not be able to recall it at the time because you're experiencing a block that prevents you from recalling it. Okay, so this again would be this is when you have something on the tip of your tongue, but you can't get to it. To handle this situation, you keep away from trying to recollect it, and you do something entirely different. And after a while, it'll just pop up into your head. And when you stop trying to search for that memory in your mind. So sometimes the information was blocked when you wanted it, and the mind brings it forward when the block is removed. So this is where stress comes into play in hindering the recall of this. This is something that you don't want to have happen to you when you're trying to take an exam. But I guess the moral to this would be, if you can't think of the answer to the question right away, go on to the next questions, and then hopefully this will come back to you while you're not focused on it. I had number 4 twice. Sorry about that. Okay, so briefly again about Alzheimer's disease. Alzheimer's is a brain disease that causes problems with memory, thinking, and behavior. So it's the most common form of dementia, but it is not a normal aging process. This is a specific disease of the brain that works on these memory systems and works on what's thought really to be part of the default mode network, and it starts to break this down over time. And if you've ever known somebody with Alzheimer's disease, the person usually progressively gets worse over time. Alzheimer's disease, unfortunately, has no cure, but there are a lot of people who research it and try to find ways to combat it, or at least, if we can't cure it, to treat it and make the symptoms of it less devastating. So I think I showed you these brains before. When I worked at the PET Center at Pittsburgh, we had people that they would do these amyloid plaque studies on to use this material called Pittsburgh Compound B to label the plaques in the brain and then see how much of this amyloid plaque was deposited in the brains of these people. So when they do a PET scan, they also do a structural MRI scan for the same reason of why we do a structural MRI when we do functional MRI. We want to, PETs are low resolution like a functional MRI. So we want to be able to co-register these two brains, line them up, and get a better idea of where anatomically the functional areas are occurring in the brain. And so these were just an MRI scan, some MRI scans that I had available to me that I wanted to show you the differences of. So on the left here is the normal brain, and on the right here are 2 Alzheimer's brains. And you can see just how intensely the ventricles of this person's brain have enlarged over time. And another thing that you can notice here is on this the far right brain, you can see where the temporal lobes, those little areas here, have really decayed and atrophied over time. So the tissue is sort of dying away or receding over time, and it's really causing a lot of memory problems for this person because remember we said that some of the memory formation takes place in these temporal lobes. Interestingly, like I mentioned before, they initially proposed and noticed that some of the areas of this decay were the same areas that were involved with the default mode network. So people were focused on that, and I believe that they still are, in trying to understand what exactly is going on in Alzheimer's disease. So again, this disease results in gross atrophy. Atrophy is sort of the decay of the tissue of the affected regions, including degeneration in the temporal lobe, parietal lobe, parts of the frontal cortex, and the cingulate gyrus. So this really is a really devastating disease of the brain. And just to conclude this memory and learning talk, Alzheimer's disease also is characterized by the presence of these neurofibrillary tangles and these amyloid plaques that I mentioned that they can now image and PET using these radionuclide tracers. So both amyloid plaques and neurofibrillary tangles are clearly visible. If you take somebody's brain from an Alzheimer's case after they pass away, you dissect it, and you can see very clearly these plaques and these tangles. And the plaques are dense, mostly insoluble deposits of amyloid beta peptides and cellular materials around and outside the neurons. And you can see the cartoon picture at the top and the actual microscopy pictures on the bottom there. On the left shows an amyloid plaque. The tangles are these aggregates of the microtubule associated protein tau, which has become hyperphosphorylated and accumulate inside the cells themselves. So they are called neurofibrillary tangles because it really does look, and this picture doesn't show it so well, but if you see these under a microscope, it really looks like the neurons, the axons in different parts of the neurons have just really become all tangled and twisted up in these brains. Okay, well, that's all I have for today about learning and memory. And I guess now we'll address any questions anybody might have, and then we're going to head over and take the exam.